Hello, welcome to another screencast. This one we'll be addressing light and pigments, the characteristics of, and some things that you will hopefully be able to identify, the different wavelengths of the visible spectrum, and the name of the pigments involved with photosynthesis, some basic stuff, and then being able to explain down here why chlorophyll looks green, why carotenoids look orange or yellow, why anthocyanins look red and or blue. Then be explaining how the absorbance or the transmittance of a specific wavelength of light by a pigment can be measured. So we're talking about light waves being absorbed as well as transmitted, and then measuring them, right, quantifying that is going to be important, especially when we go to look at the lab for rates of photosynthesis. And then finally here, how absorbance of light is correlated to photosynthetic rate. Right? We'll be working sort of on this in class, but I'm going to focus right here on these guys. So the visible light spectrum is part of the electromagnetic spectrum, and it's this narrow band right here you see of visible light. And so light has properties of a wave as well as a particle. And the part of the electromagnetic spectrum that we can see is referred to as the visible light. So wavelengths of 380 nanometers, right? So these are relatively short wavelengths here. So they're measuring from the top of one wave to the next to the top of the other, right? So this distance here, 380 nanometers, and we call that purple, violet, right? And so something beyond the violet range would be ultraviolet, right? And so these are even shorter wavelengths of light, the ultraviolet ones. Generally, the shorter the wavelength, the more, I guess, energy is contained there, the more energetic the waves are. And so really UV radiation, UV parts of the spectrum, those waves can damage cells, can damage DNA because the wavelengths are so short and there's a lot of energy there. On the other end of the spectrum, pardon me, then is red and orange, and these are longer wavelengths of light, so 750 nanometers, and then beyond that would be the infrared, right, or below the red, but really actually above the red in terms of the wavelength there. So here the wavelengths are longer, right, so from the top of one to the next. So not as energetic there, and then everything else in between. So you'll be seeing numbers like this, and again, the unit on these numbers is nanometers, right? So nano, 10 to the minus ninth, right? So this is the visible part of the electromagnetic spectrum. Now, pigments are generally proteins or protein derivatives, and they have the special ability to be able to absorb these different wavelengths of light. Now, light coming from a source here has all of the colors of the visible spectrum, right? So Roy, G, Biv, right? So red, orange, yellow, green, blue, indigo, violet. So they're all coming down there, and they are striking the pigments that are, in this case, located inside the chloroplast. And the pigment that's in the thylakoid membrane here is, of course, chlorophyll. And we'll take a look at chlorophyll in just a minute. But this chlorophyll has the ability to absorb some of the wavelengths of light, right? But then it reflects others. So the ones that are absorbed by chlorophyll get taken in, right? And so these are red, orange, yellow, the blue, and the violets. Right? But meanwhile, the green wavelengths of light are reflected, right? So they are not absorbed, right? For whatever reason, that structure of that chlorophyll molecule is not sensitive to that part of the visible spectrum that we call green. So it's not, it's here in this 530, 540 nanometer range. The chlorophyll is not sensitive to that. It's not absorbing it. And so those green wavelengths of light are then reflected. That is, they bounce off. Or some of those green wavelengths of light will then also be passed through, and we said that they are then transmitted. So transmitted and or reflected, and this is why plants look green, or most plants look green, is because the chlorophyll pigment in there is reflecting, right, the green wavelengths of the visible spectrum, right? So 540 nanometers-ish, somewhere in there, and or being transmitted. The other wavelengths are being absorbed. So here's chlorophyll. We mentioned what it looks like, so let's take a look at it. Here is, first of all, the chloroplast over here, right, outer membrane, inner membrane, thylakoid membranes, and in that thylakoid membrane, we've blown up a portion of the thylakoid membrane here. So embedded in the membrane are molecules of chlorophyll. So we take one of these molecules of chlorophyll out, and we'll take a look at it here. Now, there's no way you have to memorize the structure of this thing. Please don't waste your time or energy doing that. But what I do want to point out is a couple things about this. Right? One, there's a couple different flavors of chlorophyll, chlorophyll B, chlorophyll A, and that has to do with this functional group right here. So in chlorophyll A, it's a methyl group, and in chlorophyll B, it's the CHO group. There's a acetyl group on there. So that's a difference, and that difference translates into slightly different wavelengths of green being reflected, right? And then the other things being absorbed. So the other thing I want to point out here is that in this porphyrin ring, right, this ring is the light-absorbing head of the molecule, and then there's this hydrocarbon tail. Again, don't confuse this with a phospholipid, but it is similar to a phospholipid in that the head here is at the surface, and then the tails kind of dangle down into the middle of the membrane there, which you can't see on this. These tails here are of the phospholipids, right? Not of this. Again, let me go back and just tell you what the important part of this thing is. The important part of this thing is that there's a ton of double bonds here. I don't know if you can see that very clearly, but this is, you know, a double bond there, and there's double bonds here, and here, and here, and here, and here. And here there's double bonds, and here there's double bonds, and here there's a double bond. At the center of this thing, there's this magnesium, right? Here, this metal. And so this is an important mineral for plants to get out of the soil so they can make a lot of chlorophyll there. These double bonds, back to these double bonds, this is where the light is going to be absorbed. So when a photon of light comes in, and I'll watch some videos that show this a little bit better than what I'm showing you here. When the photon of light comes in and strikes this double bond, these electrons in this pi bond here get excited. Right? Electrons get excited to a higher energy level. So this double bond, right, when the photon of light strikes it, electrons go out, and this bond essentially breaks. So then you're left with just a single bond there. Right? So if you keep losing electrons from this thing, that's gonna be an issue. Right? But these electrons then eventually need to be replaced to regenerate that double bond, and we'll talk about what's going on in the light reactions, right, essentially there. But there's the pigment, double bonds, able to absorb photons of light of different wavelengths. Chlorophyll is sensitive to the green wavelengths of light. Other pigments, this one up here happens to be an anthocyanin. And look, cyan is blue, right, in color. So here we go, double bond, 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 double bond. So a lot of these double bonds here. And again, this is going to be absorbing, well, which wavelengths of light? The anthocyanins are blue. So which wavelengths of light are being absorbed and which ones are being reflected? This is something that you can hopefully answer there. Down here we see a carotenoid. A carotenoid, this is an example of one. And look, carotenoid carrots, what color are they? They are orange and yellowy orange, right? So if this is looking orange, you have double bonds here. Look at this thing, all these double bonds. So which wavelengths of light are being absorbed by this and which ones are being reflected? Right? So if you shine white light on there, which ones would be absorbed? Which ones would be reflected? Okay, anthocyanins absorbed here. What's going to be absorbed is red and orange and yellow and green, and what's going to be reflected, right? The blues and the violets, right? This is why they are blue. What's reflected is blue and violet. Right? Absorbed down here then would be maybe the reds, maybe not the oranges and the yellows, but the greens, pardon me, and the blues and the violets, and then reflected, and the oranges and yellows. That's why they look like that. So these other pigments here, these anthocyanins and carotenoids, are sometimes referred to as antenna pigments, or I just went out of my head. Antenna pigments or associated. No, that's not. What is it? Auxiliary. Auxiliary pigments there, right? Because in addition to the chlorophyll, which is not absorbing green wavelengths of light, like look, this one is absorbing green and this one is absorbing green. So by having anthocyanins and carotenoids and chlorophyll in the leaf, you have the ability to absorb the entire electromagnetic spectrum, all the visible light coming down on there. You're able to then grab that energy in light, all the visible light energy, and be able to convert that into the bonds in organics. So pigments are important in photosynthesis. We can measure how much light is coming through and being absorbed or transmitted. This is a meter over here, right? A galvanometer. We also will use in class a version of a spectrophotometer, right, called a colorimeter. Right? A colorimeter. And this is able to measure over here how much light is passing through a sample, how much is either absorbed or transmitted, right? That comes through the sample. So inside of the spectrophotometer, right, or the colorimeter, right, so this is in a box here, you'd have a light source, you have a prism that is able to then break the white light into the different colors of the visible spectrum. You then have this slit that's able to move, right, to pass light of selected wavelengths. So in this case, they have it lined up so that the green wavelengths of light, and again, somewhere around 540 nanometers, are passing through our sample. Right? So our sample is green, and so if this is a green pigment and we're shining green light waves on it, will that be absorbing? Right? No, it's going to be absorbing low, right? So the absorbance would be low, and the transmittance of that green would be high. So in this case, this galvanometer, this colorimeter is set up to be reading transmittance. You could also then be detecting how much was being absorbed by the sample. So in the image below here, we see that we've moved the slit now to allow blue wavelengths of light here, and so somewhere around 420 nanometers, say. And now our sample is still green, and so since it's green, it's reflecting green and orange and all those, and it is absorbing blue. So this green pigment in here is going to be absorbing blue wavelengths of light. And so you notice that the absorbance in this case, right, would be high, but the transmittance would be low. Right? So we're measuring transmittance here, and the needle is lower. Up here we're measuring transmittance, and that needle is up there at a higher range. So a spectrophotometer has the ability, the slit is really narrow, and you can move this thing up and down in terms of, like 5 nanometer differences between the different wavelengths of light there. So this is a way to measure how much a sample is absorbing or transmitting a particular given wavelength of light. This is going to be important in the lab that we do. Right? So there's information on light and pigments. And again, looking at why chlorophyll was green, carotenoids, anthocyanins, their color, how the absorbance and transmittance can be measured. And we did not get to this E, but we'll do that in class. Okay. Thanks.